topic for Chapter 9 is cell communication, or how cells send messages to each other. It's also sometimes known as cell signaling. Now, there are four main categories of signaling that depend on where the cell sending the message, as well as the cell receiving the message, are in relationship to each other. First, we have what is known as paracrine signaling. So in paracrine signaling, the cells must be fairly close together, and in fact, they must be in contact with the same extracellular matrix. In paracrine signaling, one cell releases a molecule into the extracellular matrix that then diffuses to another cell that receives that message. In endocrine signaling, the cells are farther apart. In fact, in the case of the human body, this could be a cell that's in one organ and another cell that's far away in a different organ. And the message then would have to travel through the bloodstream to get from one cell to another. Hormonal signaling is an example of endocrine signaling. Next, we have autocrine signaling. So the auto part of autocrine signaling means self. This is when a cell signals to itself as well as to other cells around it that are the same in type. And finally, we have gap junction signaling. So gap junctions, if you remember, are a type of connection between cells where they can pass things to each other. And one of the things that could be passed maybe is a signaling molecule. So these two cells obviously have to be touching and joined by a gap junction in order for this to work. So here's a diagram that just shows again those different kinds of signaling. We have autocrine signaling, where this cell releases a molecule that it itself can detect, as well as some neighbors that are maybe of the same type. Gap junction signaling, again, we pass the molecule, the signaling molecule, the message through gap junctions to neighboring cells. Paracrine signaling is signaling between nearby cells, where this cell makes the message and it diffuses through the extracellular matrix to this other cell here. And endocrine signaling is signaling at a distance, where this cell, maybe from, say, the brain, releases a molecule that goes through the bloodstream and has an effect on a target cell that's far away, say in the kidneys. So one type of paracrine signaling is neuronal signaling, when neurons within the nervous system communicate and talk to each other. The first neuron creates this message that's called a neurotransmitter that is released through exocytosis into this space here that's called the synapse. And then the second cell here has these receptor molecules that detect the presence of that neurotransmitter. And once that neurotransmitter is detected, it sets off a series of events where this cell maybe passes on the message to another cell. And that's how these messages can then be passed through the nervous system. Now, when we think about the receiving of that signal and what happens in response to that message, there are three main stages of signaling. The first is what is called reception, where we receive the message. So we have the plasma membrane here, and we have this protein called a receptor protein that's embedded in it. Now, this receptor protein detects a specific kind of signaling molecule that is meant to attach to it, like this. Now, signaling molecules are also sometimes known as ligands or ligands. You can say it either way, depending on who you talk to. So that molecule then will bind here. And when it does, it makes some kind of change to this protein that can be detected by the inside environment of that cell. This protein then will pass the message on to some other molecules within the cell, known as relay molecules, in the second step that is called transduction. Again, as the message gets passed on, typically some kind of change is made to these molecules until ultimately we reach the third stage of signaling called the response, where there is activation specifically of certain genes. So typically what occurs here then is we change the way that the cell is using DNA and maybe stop making certain proteins while we make new ones, which in turn then changes the behavior of this cell. And that is how we go from this cell receiving the signal to making some kind of change.
Now, one way that you can think of cell signaling is kind of like one of these complex machines that people sometimes build. Maybe you've built one yourself, or at least you've seen videos of it, where you put together a series of things that interact with each other, like, say, this phone knocking into these dominoes, which then rolls a ball. And the signal then is passed from piece to piece of this until in the end it accomplishes some kind of goal. So in this example, we've got the phone ringing that's slowly sliding down. That's like the cell receiving a message. And then within the cell, one molecule activates another, and we have a series of events that ends with some kind of change.
Now, the first step again of this process is what is called reception, receiving the signal. There are different kinds of receptors that cells have. And in the previous slide, we showed a receptor that was on the plasma membrane on the exterior of the cell. But there are sometimes receptors that are actually found inside of cells, internal receptors, like this. Now, in most of these cases, the receptor, once it binds to the signaling molecule, will then transit into the nucleus where it will find a particular sequence of DNA and either turn on or off the gene that's involved there. Now, in order for this to work, though, for the signaling molecule to find the receptor that's on the inside, it does mean that this signaling molecule has to be able to pass through the membrane, which again, if you remember, normally means that it is a small hydrophobic molecule. Most of the other receptors are found on the cell surface along the plasma membrane. There are several different kinds of receptors, including ion channel linked receptors, G protein linked receptors, and enzyme linked receptors. The ion channel linked receptor, as its name suggests, consists of a part that binds the signaling molecule, making it a receptor, but also an ion channel. As you recall, ions, because they are charged, have a difficult time naturally passing through the lipid layer of the membrane, and so require protein channels in order to get across. In these receptors, the channel is normally closed, but can be opened if the signaling molecule binds here and opens the channel that allows the flow of ions into the cell. Or it could be out of the cell as well. Another kind of receptor is what is known as a G protein linked receptor. Now, as the name suggests, G protein linked, there is a particular kind of protein called a G protein involved in this process. A typical G protein coupled receptor looks like this. Here's the receptor part of it, and it is bound to this other molecule called a G protein. Now, the G protein in its inactive state normally binds GDP. GDP and GTP are similar to ADP and ATP, just that it uses the guanine base instead of the adenine base. But in any case, in this particular application of it, the GDP and GTP serve to activate or inactivate this particular protein. So once again, when GDP is bound to the G protein, it is inactive. However, when a signaling molecule comes and binds to the receptor, it causes a change where the GDP gets kicked out and instead a GTP is substituted in its place. The GTP serves to activate this G protein, and when it's activated, normally it comes apart so that there is just this one subunit that goes on to initiate some other kind of response in the cell. This only occurs for so long. Eventually, GTP gets hydrolyzed, just like ATP can be hydrolyzed back into ADP and a phosphate group. And when it does, it goes back to being bound to the receptor until another signal is received to activate it again. So here's an example of a signaling event that might occur in a cell with a G protein coupled receptor. Here's the receptor again, and a signaling molecule binds to that receptor, setting off this series of events. Now, one note here, signaling molecules are also sometimes known as first messengers. So once again, once the message is bound to the receptor, it activates the G protein, which binds GTP. There are many different G proteins in cells that can do different things. This particular one goes on to activate this enzyme called adenylyl cyclase.
Adenylyl cyclase takes ATP and rather than turning it into ADP, actually turns it into a particular molecule called cyclic AMP. The M here stands for mono. So this is actually an ATP molecule that has lost two of its phosphate groups, and another chemical change makes it cyclic, circle back on itself. This CAMP is a second messenger. A second messenger is a molecule within this signaling cascade that serves to pass on and amplify the signal. Cyclic AMP in turn activates protein kinase A. Kinase is the term for an enzyme that phosphorylates something. So protein kinase A will go on to put phosphate groups onto various kinds of proteins that ultimately then initiate some kind of response. The last kind of receptor that we're going to discuss is what is known as an enzyme-linked receptor. So enzymes, remember, are catalysts that facilitate and cause chemical reactions to happen. One kind of enzyme-linked receptor is the receptor tyrosine kinase. These particular kinds of receptors actually have to dimerize. Two of them have to come together when they are activated, which occurs when the signal binds to the receptor. In this case, the receptor itself on the intracellular side contains various different tyrosine residues. Tyrosine is one of the kinds of amino acids that can be phosphorylated. We can add a phosphate group onto it. And for this particular enzyme, when that occurs, this activates it, and that addition of the phosphate group then will allow it to interact with other proteins and activate them and create ultimately the cellular response. Moving on to different kinds of signaling molecules now, the things that bind to these receptors. As I mentioned when we talked about the internal receptors, the ones that exist not at the plasma membrane but within the cell, those kinds of receptors tend to detect and be activated by small hydrophobic ligands. The reason is because these molecules that activate those kinds of receptors need to be able to cross through the plasma membrane in order to activate their receptor. And as you recall, the plasma membrane has that non-polar part of it, the lipid part, which is difficult for polar or charged or large molecules to cross through. Now, in terms of the signaling molecules that bind to the cell surface receptors, it could be a variety of different things. Now, after the signal is received, we have what is known as propagation of the signal. And in order for that to occur, the signal has to be transmitted through the membrane in a process called signal transduction. Some receptors, like the receptor tyrosine kinases, again, dimerize. They form a complex where two of them come together and work together. Within the cell, we have what is called a signaling pathway, where the message is passed through the cell. And then finally, another term that applies here, signal integration, means when you have multiple signaling events occurring at the same time, and these work together to amplify the response of the cell to these signals. So here's an example of a signaling pathway, the EGFR pathway, which stands for epidermal growth factor. Now, the receptor in this case is one of those tyrosine kinase receptors, where again the enzyme has, or the receptor has enzymatic activity such that when the signal binds, they dimerize, they come together, a portion of it becomes phosphorylated, and then that then goes on to activate other things within the cell. Notice that this one is actually also linked to a G protein here that is called RAS. So again, G proteins, when they bind GTP, they become activated so that they can go on and activate other proteins within the cell. So after the signal binds, we get the phosphorylation here. We get the activation of our G protein. That active G protein then activates this other protein that's called RAF here, RAF. And RAF is actually an enzyme which phosphorylates this other protein, MEK. Now, this protein is actually an enzyme which can phosphorylate this other protein that's called ERK. And when this last protein is phosphorylated, is transported into the nucleus,
where it initiates a change in the genes that are expressed by the cell. So you see in this signaling pathway, we call it a cascade because, again, like dominoes falling in a pathway, we have one thing that activates another, that activates another, that activates another, until we get our final response. Now, as the name of this particular pathway suggests, the epidermal growth factor, some of the end results of this is to stimulate cell proliferation or causing cells to divide and inhibiting apoptosis, which is a form of cell death that can occur. And so if this pathway gets activated when it shouldn't, that can actually contribute to cancer because then we would have cells that are dividing and not dying. Here is a video that is a good visual of a particular signaling pathway, and it's also a good review of some of the cell structures that we've talked about in the past. As we saw in that pathway, as we saw in that pathway, phosphorylation often plays a vital role in these signaling cascades. So remember, a phosphate group, we've seen this in the context of ATP before, but phosphate groups can be added to other molecules, particularly the amino acids within, particularly some amino acids within proteins, including serine, threonine, and tyrosine, the ones that have OH groups on them. In a normal serine molecule, for example, you would have this oxygen attached to an H here that gets replaced in this phosphorylated version with the P. And the same is true for these other two. Now, the name of the enzyme, the type of enzyme that transfers phosphate groups is called a kinase. There are also enzymes that remove the phosphate groups that are called phosphatases. We use the term second messengers in our discussion of the signaling cascade. Second messengers are some kind of small molecule that propagates a signal that allows a signal to be passed through a cell. Now, the reason it's called a second messenger is because the actual signaling molecule that binds to the receptor at the beginning of the pathway is known as the first messenger. So a second messenger is a molecule within the cell that helps to pass along the signal. There are a few common second messengers that we see in cell signaling. Calcium ions can be second messengers. In addition, another common one is CAMP or cyclic AMP. So again, the AMP stands for adenosine monophosphate. It's like an ATP that has two of its phosphate groups chopped off and undergoes a further chemical reaction so that the phosphate group is attached at two separate points to the sugar molecule within the adenosine, explaining the cyclic part. In order to make cyclic AMP, we do start with ATP, and then there's a specific enzyme called adenylyl cyclase, which makes this chemical change happen to form the cyclic AMP. After the cell is done with the cyclic AMP, to terminate the signal, there's actually another enzyme that comes in and undoes the cyclic part of this to make it regular AMP. There are two additional common second messengers, IP3 and DAG, which both come from breaking down a particular kind of phospholipid here. So you can see this is a phospholipid. It's got the two fatty acid chains attached as well as the polar head group here. The enzyme phospholipase C, when it's activated, will actually break this down into a couple of different molecules that look like this. This one called IP3, which comes from the polar head group here. And then diacylglycerol, DAG, which is the part that contains the fatty acids along with the glycerol. So here's an image that shows how many of these second messengers work, including calcium, which we haven't discussed yet. Calcium, if you remember from our discussion of the endoplasmic reticulum, is stored by the ER. But when certain channels that exist here are opened, calcium can flow out of the ER into the cytoplasm. Calcium, in turn, interacts with some other proteins like this one here called calmodulin, which then activates some other things and ultimately generates a response.
You can see here that this image also reviews what we talked about with IP3 and DAG as second messengers within the cell. And in fact, in this particular signaling cascade, we actually have IP3 that activates this calcium channel that allows calcium then to participate in the signaling pathway as a second messenger. By the way, even though we have a second messenger activating calcium here, we don't call it a third messenger. It is still a second messenger.
Note that this is also an example of signal integration, as you have this pathway started by the G protein linked receptor here and the receptor tyrosine kinase pathway here converging to make the second messenger IP3.
When we hit the end result response, where this is the change that is accomplished in the cell, kind of like pouring food into the dog bowl at the end of that complex dog feeding machine. In the cell, the change that's ultimately made is typically to gene expression. So we can either turn on genes so that we make protein from those specific genes, or turn them off so that we no longer make the proteins from them. So the the end of a particular pathway that's known as the MAP-ERK pathway is that you have this protein called ERK that is activated when it's phosphorylated. And so we have this ERK that's been turned on or activated. It in turn then phosphorylates some of the members of this complex here. Now, this complex is kind of like a lock on mRNA and prevents this mRNA from being translated into proteins. So in order to make the protein from this particular mRNA, which came from a particular gene, we actually have to unlock this. And so that's what the phosphorylated ERK here does. It unlocks this complex and allows this mRNA to be made into protein. So that's how genes are turned on or unlocked, or again, sometimes these pathways can lead to turning off certain genes. But what are the bigger picture results of this? What often happens at the end of cell signaling? Well, we could see increases in metabolism. So one example of that is we have the hormone adrenaline, which binds to certain receptors that sets off a signaling cascade within these cells. We see cyclic AMP as a second messenger within this pathway. And ultimately, it serves to increase the amount of glucose and hence metabolism. Cell growth is another thing that could happen as a result. In this case, we have our first messengers or our signals called growth factors, which bind to certain receptors, especially the tyrosine kinase receptors, the enzyme ones. And we see a G protein that gets activated here, which then activates that MAP kinase pathway. And again, MAP kinase ultimately unlocks the RNA from certain genes for translation. That ends up leading to increased cell division. Now, the opposite is also possible. Cell signaling can also actually lead to cell clearance or getting rid of extra cells through cell death. And this process is known as apoptosis, which is essentially a form of cell suicide. So an interesting example of this is that in the development of the vertebrate limbs, particularly the hands or paws of certain animals, the hand actually, or the paw actually starts out as one big round circular disc. And in order to form the individual digits or fingers here, organism essentially carves them out by getting rid of the cells that are in between here. And so what you see in this image is the one on the left-hand side here is a wild type or normal mouse paw that's developing. And the one that you see on the right-hand side here is a mutant where the where these cells here do not normally don't receive this apoptosis signal, and so they don't die. And what you see then in this mutant is that it develops this webbed paw, where the tissue in between the fingers gets retained. An interesting aspect of this is that there are certain animals, when you think about them, that actually have webbed paws or feet. For example, ducks. It's actually been shown that the reason that their feet are webbed is when you compare the development of their feet to similar birds, they actually don't activate the apoptosis pathway in the same way that other birds do. And so rather than developing those separated toes on their claws, they end up developing the webbed feet. The same is true when you compare mouse paws like this and bat wings, where bats' wings, if you're familiar with them, are actually a kind of webbing that exists between the fingers on their hands. And so we know that the reason it develops that way is by turning off this apoptosis pathway, which is normally supposed to clear space between the fingers or the digits here. Most of the time with these signals, we don't want them to go on forever. If you think about, for example, one of the end results of a signaling pathway could be to have cells divide. We don't want cells to continue to divide. That's actually what goes wrong in the case of cancer. So cells do need a way to turn off these signals when they're done. Now, just like we saw that kinases, the enzymes that phosphorylate molecules, often turn them on, we have the phosphatases, enzymes which remove phosphate groups that would essentially then turn these activated proteins off. In addition, as we discussed, cyclic AMP, we can convert it to AMP, which would make it no longer active in a signaling pathway through this enzyme, phosphodiesterase. And remember that calcium serves as a second messenger when we release it into the cytosol. We can actually use calcium pumps to put it back into the endoplasmic reticulum and hence end the signal that that is involved in. Most of what we talked about here applies to cell signaling within multicellular organisms, where you have many different cells that have to coordinate something between them. But signaling can actually occur in single-celled organisms, for example, yeast. A major signaling pathway that occurs in yeast is when yeast need to mate. So yeast have two different mating types, kind of like a male and female sex. And in order to be able to sexually reproduce, these two partners have to be able to find one another. So what they'll actually do is they will release these mating factors into the environment, and this mating type has a receptor that can detect the factor from the other mating type. And so that's how they find each other then and accomplish mating. This process actually has some similarities to the cell signaling that we have talked about within multicellular organisms so far, including the use of G proteins that are downstream of this, that are part of the signaling cascade. And the end result, again, of receiving a signal is to change the transcription of certain genes then that allow this mating to take place. Even prokaryotes are capable of a form of cell signaling that is called quorum sensing. So this is particularly useful in certain circumstances where bacteria, it would be of benefit to bacteria to display some kind of collective behavior, meaning that there's something that it would only be beneficial if there's a lot of bacteria around, if there's a high cell density, but wouldn't be too good if this bacteria cell is all by itself. So these type of bacteria create a signaling molecule that is called an autoinducer. Again, auto meaning self. So this is something that this bacteria cell itself can detect. Now, if this cell is all by itself, it's going to make some autoinducer that's going to and it's going to release that into the environment, but this autoinducer will quickly diffuse away. So this bacteria cell then knows that it's all by itself if it doesn't see too much of this autoinducer in the environment. If, however, this environment is crowded with many bacteria that are all producing this autoinducer, then the bacteria will detect its presence here and know that it is in a group of friends, so to speak. And when that occurs, it will change its behavior in some way. Let's say that this is a disease causing bacterium, and a part of its disease process is to make some kind of toxin that does something to damage our cells. Well, if this bacteria cell that's all by itself started making this toxin, it might quickly attract the attention of the immune system, which then would lead to it being killed off quickly. However, if it waits until there's a lot of other cells, a lot of cells of the same species around, and then they all together start producing that toxin, then they'd be much more likely to be successful since they would have strength in numbers essentially at that point. It would be more difficult for the immune system to destroy all of these cells as opposed to the one by itself. So that's one way that that has an application to human health.